Hello, and welcome to today's session on hearing. Today we're going to continue the conversation that we started in the last session where we had introduced the physical features of sound. We talked about the sound's amplitude and a sound's frequency and phase and so forth, and that was important for setting the stage about the physics of our psychological experiences of sound. But now we want to understand a little bit about the physiology before we can more fully understand the psychology of our auditory experiences. So today we're going to be talking a lot about the anatomy and the physiology of our auditory system. So we'll jump right into it and see if we can set the stage first at a relatively large level, and then we'll begin to zoom into different components of our anatomy and physiology with respect to hearing. So here's our introductory photograph, and what we have is a very large-scale picture of the earliest portions of our auditory system. We'll start out here with the ears, and we have the outer portion or the visible portion of our ear, which we refer to as the pinna. And the pinna helps to funnel some sound into the auditory canal. And then once the sound has gotten into the auditory canal, it eventually bangs against our eardrum. We talked about how sound is a physical wave. It happens to be a longitudinal wave that'll be banging against our eardrum, which is sometimes called the tympanic membrane. And then from there, we're going to see some finer structures in the middle ear, which is also an air-filled chamber. So out here where the sounds are bouncing around in the real world, typically those sounds are bouncing around in air. Inside of this middle chamber, the middle ear, we also have air, and so that's an air-filled chamber. And then we're going to have a structure that we call the inner ear, and we're going to spend a lot of time today talking about cochlea. Some of you might know about cochlear implants. The word cochlear is the adjective form of the word cochlea, and we're going to spend a lot of time today talking about cochlea and how they are comprising different kinds of membranes that allow us to hear. So in big picture, we have this diagram, and we might even schematize it this way. We might talk about the outermost portions of the ear, the pinna and the eardrum. These act more or less as directional microphones funneling the sound into a chamber that can subsequently set up the transduction, converting the physical energy of sound into some kind of physiological response, eventually turning this into a neuronal response. Okay, so that is setting that up as a directional microphone. In the middle ear, which we've mentioned is air-filled, we have a couple of functions that we're going to have to unpack. Some of this terminology will be new to you. We're going to do something called impedance matching, which we also call resistance matching, and something else called overload protection. And you'll see in a few moments why those are such important functions of the middle ear. And then later on we'll talk again about the cochlea, which we call a portion of the inner ear, and we'll talk about something called a frequency analysis. We've mentioned a few times in this course that we have Fourier analyses done throughout our nervous system. It appears to be the case that our occipital lobe performs something like a Fourier analysis on the visual images that are landing on our retinas. It also appears that portions of our auditory system perform something akin to a frequency analysis, and this will be seen most saliently perhaps inside of the cochlea in the inner ear. So why don't we begin to zoom in on different portions of this. Okay? We'll talk first about the outer portions and then the middle ear. Okay? So this will be the sequence of anatomical structures that would set up the phenomena of hearing. Okay? So we'll talk first about outer and middle ear. So the outer ear, as we've already mentioned, comprises the pinna, the auditory canal. This is what you typically are cleaning when you're using Q-tips to clean your ears, the auditory canal, and then the eardrum. You don't want to go in too far with your Q-tip, or you might bang against your eardrum, which we also call the tympanic membrane. And that's fairly easy to remember. Many of us are familiar with these big kettle drums that we call tympani, and so if we think of the eardrum as a tympanic membrane, that would be a helpful mnemonic. Right? After we get past this outer ear that has these three components, including the eardrum, which is going to be vibrating more or less like a barometer, might be registering the different changes in barometric pressure. We talked about how we have compressions and rarefactions in our last section. So now we have the tympanic membrane or eardrum wiggling back and forth in response to those air pressure changes. And then we come up to the middle ear. As I've mentioned before, this is an air-filled chamber between two membranes. And in today's session, and also in class, we'll be talking about how these membranes bound the middle ear. On the one side, I'll make that on the left side for you, we'll have the tympanic membrane. On your right side, we're going to have a much smaller membrane that will be called the oval window. And in between the oval window on your right and the tympanic membrane on your left, we have this air-filled chamber. Okay, and that's going to be our middle ear. Okay? The middle ear contains 3 of the smallest bones in the body. This is actually, I think, one of the great feats of evolution, that evolution would give us these bones that work so intricately together. These are called ossicles. And there are 3 of them inside of this air-filled chamber that we call the middle ear. They go by these names, which are, I believe, Latin names, malleus, incus, and stapes, and that is the sequence in which they would occur from the tympanic membrane. We first encounter the malleus. It's attached to the eardrum, or tympanic membrane. That is going to be connected by ligaments to the incus, which we might think of as the anvil. It's bounded to this and also to the stapes, or stirrup, the next bone and the final bone in our 3-bone chain. So very tiny bones working together to form something akin to a lever system. And we'll see why the lever system is very important. One note about the stapes, it's bound to and strikes against the oval window. That's going to be over here on your right-hand side in our example. Again, we have the tympanic membrane over here, the oval window, and then we have these 3 ossicles that are going to form something like a lever system, malleus, incus, and stapes. Over here, on the within the stapes, we have these connecting ligaments to the oval window. And to use a very crude analogy, it's almost as if the stapes bangs against the oval window in a manner that a drummer's bass drum pedal bangs on the bass drum. Some of you have seen drum kits that have a foot-operated bass drum pedal, and that's banging on the bass drum to give us a beat. A similar kind of principle is going on here with the stapes banging on the oval window to set up some perturbations inside of the inner ear, and we'll talk about that in just a moment. So let's see if we can understand a diagram of the middle ear. This again is air-filled. Okay, here's the big picture. Right, here's your pinna coming through the auditory canal. Here's where it starts. We have the oval window, the tympanic membrane, or the eardrum. And if we zoom in, here's the eardrum again, or the tympanic membrane. And then we have our hammer, anvil, stirrup, malleus, incus, stapes. And then this is where we have the stapes banging on the oval window. So hopefully that gives you some idea about how this is laid out. Okay. What does the middle ear do? We mentioned in our opening slides that it has a couple of functions. We'll see if we can understand some of those because there are some interesting physical concepts going on here. One function of the middle ear is overload protection. Overload protection is granted by the acoustic reflex, which stiffens the ear and restricts the ossicle's movement. So let's see if we can understand what's going on there. As I'm speaking to you on this video, I'm putting out quite a bit of acoustic energy through my vocal apparatus. My vocal cords are flapping, and actually, if you look at the distance between my mouth and my ears, it's quite small, and that would imply that the sound coming out of my mouth should be fairly loud at my ears. And yet I don't damage my ears by speaking, and neither do you. Why is that the case? Why can the ear be so proximal physically to such a loud source of sound and yet never deafen itself? The answer is that we have this acoustic reflex. This is not something we have to practice. This is something that happens all the time. When you and I go to speak, when we go to chew or swallow, there could be a lot of acoustic energy right here at our ears, but what we do is momentarily make our ears less sensitive by tightening the muscles that control this lever system that we call the ossicles, the hammer, anvil, and stirrup, the malleus, incus, and stapes. Those are bound by some muscles that tighten up and make the ear very momentarily less sensitive, and they protect the ear from overloading. Overloading could have been a tremendous problem when we're speaking, could have been a tremendous problem when we're chewing. I have a colleague, I won't gender this person, and he or she has a tremendous sneeze. A lot of acoustic energy comes out of that. That could have damaged the ears, but we have overload protection, okay, provided to us by this acoustic reflex. Okay? And when we return in a few sessions to talk a little bit about echolocation, we'll see how the acoustic reflex plays an important role in bat echolocation. Okay? So that's one of the major functions of the middle ear, is to make sure that these ossicles don't get overloaded when we're speaking, when we're chewing, when we're sneezing, and so forth. We want to prevent the ear from damaging itself. We have to prevent ourselves from shooting our ears out. Okay, so that was one of two functions of the middle ear. Let's see if we can now understand this other function that we call impedance matching. I mentioned a few slides ago that our synonym for impedance is resistance. Okay, so we're going to talk about resistance matching. And just to give you a little bit of an analogy, I'll help you think about resistance matching or impedance matching in this way. We've been talking about how this middle ear is an air-filled chamber. It contains air, just like the air out here in the real world. When we get to the point where the stapes is banging against the oval window marks the beginning of the inner ear, and the inner ear is fluid-filled rather than air-filled. Okay, so we have air-filled here in the middle ear and a fluid-filled inner ear. Middle is air, inner is fluid. Okay, so why would that matter? Let's see if we can understand why that's related to this issue of impedance matching or resistance matching. We can think of light as a messenger. We can think of sound as a messenger. These messengers are conveying information, the reduction of uncertainty, about distal stimuli to us here on our central nervous system or our peripheral nervous system. Okay, we call that the proximal stimulus. Okay, so we have these different messengers, sometimes light, sometimes sound. Imagine that you're a messenger, and your job is to convey a message by carrying it for 100 yards or 100 meters. And let's say for the first 100 meters, your job is to carry that message through the air. So you're walking on ground like you normally do, and you're just moving through the air. Nothing particularly unusual about that. But for the remaining 50 meters, your job is to carry that message through water, and you're asked to now walk or run through the water. If you've ever done that in a swimming pool, or in the ocean, or in a river, or in a lake, you know that there's much more resistance in this fluid-filled chamber that might be the pool or the lake than there is when you're walking through air. So air provides some level of resistance, but fluid provides a much greater level of resistance. So if we're going to have a messenger-like signal propagating across air-filled media, like the middle ear, and also fluid-filled media, we have to be prepared for different kinds of resistances, different kinds of impedances. And we're going to try to match the impedances as best as possible so that we're not losing any kind of information when we get to some resistance from a, if you will, thicker medium, like a fluid-filled medium. Okay? So the fluid-filled inner ear has more resistance than does the air-filled middle ear. So sound would be lost if it were not amplified in some particular way inside of the middle ear. So we need the middle ear to protect us from overloading. We also need the middle ear to amp up the signal in some way because it's going to take us right into a fluid-filled medium where there is more resistance. So amplification now, this impedance matching, is going to be achieved by the ossicles and by forcing the vibrations from the relatively large tympanic membrane on this side to a much smaller oval window on this side. Okay? Let's see if we can understand each of those two components. Okay? The ossicles form something of a lever system. And you might know how levers work. We can get leverage in lifting relatively heavy objects by taking advantage of levers. And you might think of a seesaw as something akin to a lever. And I always like to tell this story. When I was in grade school, I had a really good friend. His name is Neil McHugh. And Neil was much bigger than I was, even though we were the same year. I was among the smallest people in the class. Neil was among the tallest. And so we would occasionally be together on the seesaw, and you can imagine that we had a bit of an imbalance. But what you can do is we can have the larger person move closer to the focal point, and you might be able to achieve a balance that way. The point about all this is that levers can help us increase our strength in lifting a particular object. Levers can increase the energy within a system, so to speak. Not formally speaking, but it can amplify a sound. And that's really what we want to think of the ossicles as doing here, amplifying a sound. So we have this lever system that's going to be transducing some activity here on the tympanic membrane, and through this lever system down to the oval window, we're going to be amplifying the acoustic energy that's coming in. Separate and apart from that lever system that is created by the ossicles, we have another imbalance that's actually working to our favor. Imagine that we have a certain level of resistance or a certain level of energy, I guess we'll call it, out here on the tympanic membrane. Okay? And we have a much smaller surface over here on the oval window. The difference between that size and this size tends to work for us. We're taking the same amount of force and we're now putting it into a much smaller space over here on this side. Okay, so one of the ways to think about that, and I use this analogy every year in class, is imagine that you're walking across campus and it had been raining fairly lightly in the recent hours, and so the ground is a little bit wet. If you're walking barefoot across campus in that relatively moist ground, your heel will sink down just a little bit, but not too much as you're walking across. Your heel has a certain width and a certain expanse of area. Right? Imagine, though, that instead of walking barefoot, you're walking across the same surface, but you're wearing stiletto heels. Now you are the same weight as you were a moment ago, but all that weight is being pushed onto a surface area that's much finer than would have been the case for your barefoot or your heel. Okay? So now as you're walking across, you're going to be digging down into the surface because you have the same amount of weight, but you're applying that to a much smaller surface. Okay? And that is the same analogy that we have with this relatively large tympanic membrane on one side coming down to a much smaller oval window. So we have a couple of ways of handling the impedance difference between the air-filled middle chamber and the fluid-filled inner ear, starting with the cochlea, beginning at the oval window. Okay, we have this change in size from a much larger size. Same level of force is now being applied to a smaller area. And we also have the lever system that's set up by the ossicles. Okay, so now we'll talk a little bit about the inner ear. And this is the cochlea that we're going to be addressing here. The word cochlea is from the Greek word snail, and you can see it's somewhat snail-shaped. The cochlea is approximately the size of a pea. So all of this talk that we're going to have about cochlear implants, we're talking about structures that are something not bigger than the size of a pea. A lot of biological complexity here in a very small region of space. So we're going to uncoil this snail-shaped structure, and we'll talk about portions of it. We won't do all of the cochlea, but we'll talk about those most relevant for hearing. Okay? So here's the cochlea in snail shape. We're going to now unravel it, and we'll take a look at it this way. And we're going to limit our discussion to a couple of structures here. The organ of Corti. This is named after the anatomist, the Italian anatomist Corti, who first discovered this and described that membrane structure. And then we have also the basilar membrane, and we'll talk about how these two are going to play an important role in the anatomy and physiology of hearing. Okay, so the organ of Corti and basilar membrane are the two that we're going to focus on. The organ of Corti, if you will, sits on top of the basilar membrane, and I'll ask you to do this with your arms. So the one on the bottom is going to be the basilar membrane, the one on top is the organ of Corti. Okay? And there's a nice mnemonic here. The basilar membrane is on the bottom, and the word bottom has a B and an M in it. So that's a handy little mnemonic for understanding which one is where. More specifically, the portion of the organ of Corti, which is the one on top, that makes contact with the basilar membrane is called the tectorial membrane. So here's the basilar membrane. On top we have the organ of Corti, and on the bottom of the organ of Corti is a particular membrane called the tectorial membrane. Okay, so the tectorial membrane and basilar membrane slide back and forth across each other, bending the hair cells that are in between those, and we'll zoom in and talk about those in a moment. And these hair cells are going to be the receptors for hearing. Just like we have photoreceptors in vision, we have these hair cell receptors for hearing. Okay? This is the way I typically offer a gesticulation for this transductory process, this conversion of some kind of energy, maybe physical energy or mechanical energy or chemical energy, into action potentials. And this is where it's beginning to happen inside of the cochlea with the sliding back and forth of these two membranes. So I'll ask you to put your arms like this and slide back and forth. Okay? This is analogous to the other gesticulation we had made when we studied the visual system. We talked about the cis-trans isomerization. What's interesting about that is that's actually a chemical change. It's a conformational change from the cis form to the trans form of the relevant molecules. Okay? Here what we have is not chemical energy, but rather mechanical energy. As we have the stapes banging on the oval window, creating a rhythm, so to speak, in the fluid-filled inner ear, we're setting up some waves, and the waves are going to differentially move the organ of Corti on top and the tectorial membrane right underneath it against the basilar membrane. And as these are going back and forth, we're going to have these hair cells that are going to be moving back and forth, and they're going to be releasing neurotransmitter. So the bending triggers electrical changes in these hair cells, and this generates a release of neurotransmitter. So all of a sudden now we have what we're typically used to seeing, the release of some neurotransmitter, and this is all done through a very mechanical process, unlike the chemical process that we had over in vision. So this is how the two systems might differ. This is very mechanical in hearing. Vision was very chemical in its origin. Okay? All right, so it's a little bit like this transisomerization, but not exactly the same thing. Okay, let's see if we can talk about some facts relating to hair cells, the receptors for hearing. Hair cells are the receptors for hearing, and they come in two varieties, inner hair cells and outer hair cells. These are not perfectly analogous to rods and cones, but what is nice here is that there's a little bit of a similarity. We know that we have different kinds of photoreceptors, rods and cones, and within the cones we might have different varieties. Very crudely, the red, green, and blue cone types. We know why that's crude and why it's inappropriate to call them red, green, and blue, but we have different varieties of these receptors. Same thing is true here. Instead of having rods and cones, we have inner and outer hair cells. In each ear, there is one row of approximately 3,500 inner hair cells, and 3 rows of outer hair cells that total to about 12,000 hair cells. So you can see that we're dealing with receptors on the order of thousands rather than on the order of millions, which is what we had in the. We actually had maybe more than 100 million photoreceptors in each of the eyes, more than 100 million. Here we're down to something on the order of, all combined, maybe 15-ish thousand cells for hearing. And both types of hair cells contain bristle-like structures on their tops, as shown here. Okay, so this entire structure is the hair cell. Here's another hair cell, inner hair cell, outer hair cell. We have these small bristle-like features on top that we call cilia, and cilia would be part of either inner hair cells or outer hair cells, and the bending back and forth of those cilia will generate the release of neurotransmitter that will set up the signal that's going to make its way through the thalamus and off to the auditory cortex in just a few moments. So that's where the action is in these cilia. We're always concerned about damage that is done to cilia when we're listening to sounds that are really, really loud. Okay, I love this photograph. This is something that was taken by way of a microscope, and we can zoom in and see the beautiful biological organization and the biological complexity of outer hair cells, OHC, and inner hair cells. And we can zoom in even further and see just how beautiful these look, inner hair cells and outer hair cells. You might notice that we have these 3 rows for outer hair cells and just 1 row of inner hair cells. They're also in this beautiful U shape for the outer hair cells. So all of this is going on inside of our cochlea. Really some nice looking pictures here. Okay, some other facts about hair cells. Even though outer hair cells outnumber inner hair cells, the inner hair cells enjoy something that I jokingly refer to as most favored hair cell status. Some of you who've studied political science might know that there's something called most favored nation status. And this might be a status that one country has with another to set up some kind of trade privilege, something along these lines. Well, it turns out that not all hair cells are created equally. Not all of them are equally influential in transducing acoustic energy and sending a signal off through the thalamus to the auditory cortex. Right? It looks like the inner hair cells are going to be steering the ship most of the time here. That is, of the 30,000 auditory nerve fibers, so just like we have fibers coming out for the visual system coming out of making up the optic nerve, we also have the acoustic nerve. And this is going to be the 8th cranial nerve, by the way. So the optic nerve was the 2nd cranial nerve. The 8th cranial nerve is going to be the acoustic nerve. And there will be 30,000 auditory fibers that are carrying signals there between the cochlea and the brain. Only 5% of these fibers are linked to OHCs, okay, the outer hair cells. So a relatively small fraction of these fibers are carrying information relevant to the outer hair cells. So 95% of the auditory nerve takes its orders from the inner hair cells. Okay? So inner hair cells are more or less steering the ship. Although the outer hair cell activity makes a comparatively small direct contribution, outer hair cell activity makes a large indirect contribution by affecting the output of IHCs. So there are a couple of analogies here. I think the one that I like the best is that you might think of it this way. To a first approximation, the inner hair cells are something like the members of Congress. The members of Congress actually get to vote, and the votes are counted. The outer hair cells are better analogized to lobbyists, and we might have many, many lobbyists for any given inner hair cell. So the actual vote comes from the Congress member, and the lobbyist is now going to be trying to influence that vote in some way. There's some very interesting recent information that's even more recent than what you have in your book that suggests that what the outer hair cells primarily do is they take top-down signals. That is, if you can imagine that you're trying to use your memory or attention or something that's driven by some relatively later portion of your nervous system, and you're trying to influence what it is that you're hearing. Maybe you're going to try to zoom in on one sound over another sound. The outer hair cells can help with that process. They tend to modulate the bottom-up signals by taking top-down information from later portions of the brain back to the hair cell level. Okay? So they're trying to, these are engaged in influencing the activity of the inner hair cells, often by top-down signals. Very interesting recent finding. Okay. There are two main ideas about the inner hair cell activity and how it codes frequency. So why don't we go on to talk about that now? Okay. There's a temporal theory. And the temporal theory originally posited that higher frequencies are associated with greater number of action potentials in the cochlea. So let's talk about the range of frequencies that we can hear and how we might code that inside of our nervous system. So as you might recall, we talked about how we have moments of compressed air and moments of decompressed or rarefied air. And these cycles of compressed versus decompressed air are happening many times per second. The human auditory system can typically track a range, and I hope you'll make note of these numbers, that goes from about maybe 16 hertz or 16 cycles per second to something like 22,000 cycles per second. That's the range that you and I can typically hear. Okay? As a matter of fact, that's probably the range that you can hear, but as we age, we tend to lose some sensitivity to the higher frequencies. So people your age typically can detect 20 micropascals, and your frequency range would be something like 16 hertz to 22,000 hertz. Okay? All right, so if you can imagine then that we have the auditory stimulus coming in and it has different kinds of frequencies, one simple way to think about it is if I get something like 16 cycles in a second, then I might have something like 16 action potentials. If the frequency is increasing and it goes up to 10,000, or we'll say 100 cycles per second, then I get 100 action potentials per second. So there could be a one-for-one mapping, was the idea in the temporal theory of how frequency is coded in our nervous system. Now there are some limitations with this, and one limitation with the theory is that each neuron will have a refractory period of about 1 millisecond. And you might remember from Introduction to Psychology that we do have these refractory periods, an interval of time during which the neuron cannot fire. It's engaged in resetting its membrane potential and hyperpolarizing itself, or at least polarizing itself, so that it can set itself up for the next action potential. Well, that refractory period is going to render the neuron unable to fire for some interval of time. And typically, a neuron in the mammalian visual system or auditory system would not fire faster than 1,000 times per second, and often it's a lot slower than 1,000 times per second, or 1,000 hertz. So we know that an individual neuron can only crank up to about 1,000 times per second, and yet we know that behaviorally, you and I can hear sounds that range from 16 hertz to 22,000 hertz, and that's quite a range and goes beyond the speed of any individual neuron. So one of the ways to circumvent this problem from the early ideas about the temporal theory was to say that maybe there is something like a volley, that we have these neurons that are going to work in comparison or in cooperation with one another, and that they're going to operate collectively by volleying one across the next. So the volley theory posits that a group of neurons could have interleaved responses that could achieve rates greater than 1,000 hertz. So here again, sometimes a picture will say 1,000 words. This might be our first neuron, and it's firing away at some rate. Here's our second neuron, also firing away at some rate. And either of these can only fire at something like, we'll call it 1,000 times per second. But if we have them work together in a volley, then maybe we can get twice as many per unit time if they're appropriately spaced in time. And we now have this interleaving one with the next, and you can see that fibers A and B collectively are carrying more action potentials than any one of these can carry by itself. So here I'm using 2, but you can imagine now factoring in a third one. And they now are taking turns with each one of these taking 1/3 of the interval. And so we can then add a fourth one and a fifth one and so forth. It's one of the ideas about how you could get to a 20,000 hertz coding using only neurons that are individually limited to just 1,000 hertz. Okay. Some people still have questions about the volley theory. It might depend on a neuron that's supervising all of that activity. That is to say, if we have a group of neurons that are volleying like this, how do we still extract from that this higher rate, this now 2,000 cycles per second rate? Do we need some other neuron that's reading these neurons? And if so, that neuron itself would only be limited to 1,000 hertz. So it's not entirely clear that we can get to these really large numbers of cycles per second, like 22,000, by using any kind of volley mechanism. So temporal theory and volley theory could be true, and they probably are true, but most likely they're encoding frequencies that are on the lower end of our frequency spectrum. For the higher frequencies, we need to posit some other kind of a theory, and what we typically use is what we call a place theory. Okay? So these two theories can work in conjunction. They're not mutually exclusive, and the place theory is very nicely analogized to what we see here. You can think of a piano going from low to high, and that the higher notes are arise from a string that's relatively shorter and therefore has generates a shorter wavelength, which is a higher frequency. The lower notes have these longer strings that will generate longer wavelengths, which will have lower frequencies psychologically. So the place theory holds that a traveling wave of excitation is maximal at different places on the basilar membrane. Okay? So here's one of the ways of thinking about it, at least metaphorically. This can be our basilar membrane. And you might see a similarity between the length of these individual strands and the length of the individual strings inside of a piano. This was actually Helmholtz's early idea about how the basilar membrane might encode different kinds of frequencies. And he had this notion that we might have relatively low frequencies down on this side, and here at the base, this, by the way, is where the oval window is, and where the stapes is banging against the oval window, we might have relatively high frequencies positioned there. As it turns out, we really don't have strings or ligaments moving across the basilar membrane as Helmholtz had originally thought, but Helmholtz had an important insight, and that is that the place or the location of maximal displacement along the basilar membrane will have a correspondence with the pitches that we hear, and there's a really nice demonstration of that by a researcher named Bekeshy. Right? And as you might remember reading about, what Bekeshy had done was he had taken this metal tube and he sliced it. And after opening up the metal tube, he laid across the metal tube a rubber membrane that had the same kind of broadening that we see anatomically when we take a look at somebody's basilar membrane. The basilar membrane starts narrow-ish and it widens as we move toward its apex. So he did that inside of this now halved metal tube, and he's got this rubber strip coming across, this rubber membrane. Then he closes it up, okay, and he begins banging on one side of it, just like the oval window is banged upon by the stapes. And you can rest your arm right on top of that device, and you can begin to feel which portions of this tube are now going to be vibrating maximally when you have a tuning fork that's banging against this thing at a relatively high frequency or at a relatively lower frequency. And he demonstrated this proof of principle, which is the idea that we get maximal displacement in a region that corresponds to the frequency of the stimulus. Okay? So a very nice demonstration here in the Bekeshy study. And then we can see how that might be more related to the actual structure, which is curved. We had unpacked it and unraveled it for understanding it, but the actual device, the actual cochlea, is a snail-shaped spiral, and we can see that we might have very high frequencies encoded near the base And then as we move, it gets broader and broader and broader, and we have lower frequencies represented further away toward the apex. Okay, so low frequencies at the apex, high frequencies at the base. Here's something that I hope will help you to remember some of these details. There are two spellings of the word that we pronounce bass. Okay? So here's the word bass, B-A-S-S, and this typically refers to a bass instrument, like maybe a bass guitar or the low end of a piano. Okay? And we also have this word bass, which will mean the bottom of a structure, okay? Like the base of a table or the base of a desk. Okay? So what we might remember is that there's a backwards relationship here. Backwards because the base is not the base. Here's where the oval window is, and we have the stapes banging across that, and we have very high frequencies here. And as we move away from the base, B-A-S-E, we start having maximal displacement at different areas until we get the lowest sounds, the bass sounds, if you will, from the bass guitar, B-A-S-S, now are being represented furthest away from the bass, B-A-S-E. Okay? So it's backwards because the bass, B-A-S-E, is not bass, B-A-S-S. I hope that helps you. Okay, here's another picture shown in a slightly different orientation. Here's the oval window. The stapes, one of the oscillacles is banging on that. We're going to set up a traveling wave of disturbance across the basilar membrane. Here's the apex, and you might get a point of maximal displacement that is going to be representative of what sound or what frequency had been banging against the oval window. Okay? So here we have two different stimuli across time. So here's time 1, 2, 3, 4, and 5. And as time goes on, we get a increasing wave size typically until the wave finally subsides. And here we have a response to a high frequency tone. Okay? So in the high frequency case, we're getting a maximal wave relatively close to the base, B-A-S-E. Okay? And over here we have the response at the basilar membrane to a lower frequency tone. Okay? And so here you can see that for the lower frequency, here's the base and here's the apex. We're getting maximal displacement closer to the apex than we are to the base. So low frequency versus high frequency changes in time as we go down. I think this is a very helpful picture in understanding this moderately complicated structure that is the basilar membrane. Okay, the auditory nerves make their way to the medial geniculate nucleus. So we've talked a little bit about in vision how we have the optic nerve, which is the second cranial nerve, make its way out and go through the thalamus, specifically the lateral geniculate nucleus, all the way back to the primary visual cortex. Now we're going to come to a roughly analogous sequence of events. Now we've got the auditory nerve, which is going to be the eighth cranial nerve, making its way again toward the thalamus, but instead of going to the lateral geniculate nucleus, we're going to go to the medial geniculate nucleus, okay? And then make our way off to the primary auditory cortex, which will be signified by A1 for auditory, as opposed to V1 for visual. Okay? So neurons in the MGN, or medial geniculate nucleus, project to the primary auditory cortex, and these will be located in the temporal lobes, one on each side. So similar to LGN to V1, we now have MGN to A1. And A1, and in the superior olive, a structure that's occurring earlier than the cortex, some neurons receive input from both ears. So we talked about how in vision, we have individual cells that might be in the cortex, and they receive input from the left eye and from the right eye. It turns out that in the auditory cortex, area A1, we have cells that are also binaurally innervated, which is to say they receive input from the two ears, and there are even some earlier structures, including the superior olive, that have that feature. These are called binaural neurons, and they help to localize sounds. And we mentioned the other day that when we were talking about the phase of a sound is going to be playing an important role in how we localize that. Okay? So let's see how a binaural neuron might begin to engage in some kind of localization. First, just to set the stage for this, this is one of the few things that we can do when we're born. Assuming that a newborn infant has been developing in the womb for, I would say, at least 8 months, but if you get to the 8-month mark, or maybe even earlier than that, when the baby is born, the baby can localize to sounds immediately. You don't have to learn how to do this. This is true for many different animals, not just for the human animal. If you snap on one side of a baby's head, assuming that the baby has anywhere near normal hearing, the baby will orient it to it immediately. You can snap on the other side, the baby will orient over there. So even within the womb, we have some cortical wiring going on that will allow for this sound localization. And let's see if we can understand here how the binaural neurons might be wired up. We'll take the case over here where we have a neuron, a binaural neuron, we'll call it A, that registers sounds from the left. Okay? So here we have a sound coming in from your left-hand side, and that sound is going to hit this ear first, and subsequently, maybe a small fraction of a second later, it will hit that ear. So the time of arrival is first here, and then secondarily here. Okay? So we're going to have the sequence of events that we've talked about in each of those ears. We're going to get some wiggling on the basilar membrane and the tectorial membrane, and we're going to have the release of some neurotransmitter, and the sequence of action potentials will begin making their way toward a binaural neuron, for example, in area A1. Now, because this ear received the information first from the real world, and this received it second, you might imagine that this signal would get to the binaural neuron first, and this one would get there second. Okay? So what we do is we have neurons that actually are looking for the coincidence of these two, but along the way, we might add a small delay, delta t, to the first one, such that this neuron will only fire when these two feeds come into it at the same time. So we might add a small temporal delay that would now offset the fact that out there in the real world, the left ear was stimulated before the right ear was, and then this neuron will fire, and when that neuron does fire, you have the psychological experience of having leftward positioned auditory stimuli. Okay? Let's do the same thing over on this side just so it's really clear. In this case, the sound is coming from this direction. Okay? It's coming from your right. It will hit the right ear first, so the right ear is off to the races. Then it hits the left ear. The left ear is getting a bit of a delayed start. But we can compensate for that delayed start by adding some kind of delay in the right ear's feed, such that when these two signals are now arriving at the binaural neuron, they arrive at the same time. Again, we call these coincidence detectors because they are marking and responding to the coincidence of these two feeds. And when these two come together, this neuron will fire, and when it does, you have the psychological experience that there was a sound on your right-hand side. So all of this wiring was going on even in your womb, and at the time of birth, most typically hearing babies have no trouble localizing sounds. It's interesting to muse about how the auditory system could build something like this. It might be the case that we create a delay here by having a slightly longer axon so that the action potential has to go a longer distance to compensate for the fact that this ear received the signal first. It could be that rather than elongating the axon, we have maybe lower levels of myelin. Okay? Myelin tends to help the signal move faster. We can slow down this side by having lower levels of myelin. We can also leave the same amount of myelin there, but we can have the quality of the myelin degraded. And there's a very interesting technique now called diffusion tensor imaging, or DTI, that measures the quality of fiber, the rate at which water molecules will flow down the fibers. So we can have a qualitative difference in the fiber. We can have different amounts of fiber. We can have different length axons, different ways that we could possibly build these feeds into the coincident detecting binaural neurons. You might recall that in the earliest portions of our course, we talked about David Marr's 3 levels of analysis. There's the computational level, we map inputs to outputs. There is the algorithmic level, we say mathematically how it is that the inputs are mapped to outputs. And then we have the implementation level. And here we're talking about different ways of potentially implementing a binaural neuron, maybe by having longer axons, more or less well myelinated axons, and different kinds of myelin quality would do the trick at the implementation level. Okay, just as V1 is retinotopic, area A1 is tonotopic. Okay? So you might remember that in area V1, as you marched across from one cell to the next, you had a nice representation of what was going on out here on the retina. In area A1, as you march across with your electrode, you'll notice that you have a tonotopic map corresponding to different positions on the basilar membrane. So positions of various frequencies represented in A1, the auditory cortex, correspond to positions back in the basilar membrane. Let's see how our sensitivity to various frequencies and intensities might depend on the response of individual auditory neurons. Okay, so here we're showing you the schematic for the behavior of an individual neuron that might be an auditory neuron in area A1. And what we have on the x-axis are different tone frequencies, from relatively low tones to relatively high tones. If I could whistle what this might sound like, if I were to go from here back this way, it would go from lower tones to higher tones in the frequency domain. On this y-axis, we're reporting now something related to decibels. Remember that we talked last time about decibels? This is the intensity of the sound that we need to make this neuron fire. So if we have lower and lower intensities that make this neuron fire, that means this neuron is getting more and more sensitive. So on this axis, we can think of lower scores as being better or more sensitive responding. So here would be the amount of energy that we need at different frequencies in order to get this neuron to fire at some specifiable rate. So at these relatively low frequencies, this neuron will fire, but we need a lot of acoustic energy. We need many dB SPL in order to make this thing go. But as we get out to maybe 5,000 hertz, we can make this neuron begin firing even at relatively low intensity levels. So this neuron then is more or less tuned to this particular frequency. It has a characteristic frequency, in this case, of 5,000 hertz. Some other neurons will be tuned to higher frequencies, others to lower frequencies. Okay, let's see if we can also get to intensity coding. And I alluded to this last time when we had our conversation about pressure levels and intensity levels and so forth. Here we're going to show two different neurons. And here we have sound intensity now in dB SPL, that's sound pressure level, on the x-axis in each case. And we have the strength of the neuronal response on the y-axis in each case. This is a high level of firing. This is a low level of firing. High and low again. Here we have a relatively sensitive neuron. Okay? It's going to have some spontaneous level of activity. And then at some threshold level of intensity, maybe something like 20 dB SPL, this neuron starts firing monotonically. That is, it increases its firing rate with increasing sound intensity until it saturates or reaches asymptote. Okay? And then no matter what we do beyond that, this cell will just continue to fire, but it can't fire more than its maximal rate. So this can encode changes in intensity over this range, but not beyond that range. And it kicks in at about 20 decibels. Okay? We also have other neurons that are going to similarly show spontaneous levels of activity, but they won't kick in and start firing monotonically until some higher level, maybe 40 or 45 dB SPL. They begin to respond with increasing levels of firing to increasing levels of sound intensity as measured by dB SPL. They too will eventually hit their own asymptote and not be able to differentially encode any additional sound intensity. Okay, so that is our introduction to the anatomy and physiology of the auditory system. We hope that, as always, you'll jot down what was particularly clear about this lecture and what was less clear to you, and we hope that you'll bring your questions and comments to class. See you in class.